known as PEMRIG for short. Our aim is to bring you the latest research information on Parkinson's. Our expert speakers will be providing their insights into the most important developments and research to improve the treatment of Parkinson's. Parkinson's Research Support Network brings together people driven to help find a cure and better treatment for Parkinson's. Everyone can play a part in the search for a cure and better treatment for Parkinson's. The more people who get involved, the faster we'll get there. It's great to see you, and I'm looking forward to the presentations by our three excellent speakers. It's good we have been able to invite people throughout the country this time, as it's online. I've just learned that we've got 70 guests, which is great. I hope you will have lots of questions for individual speakers or for the panel session at the end of the meeting. If you have a question, please use the chat facility and we will do our best to arrange for it to be asked on your behalf. Liz Nash, Research Support Manager, is helping me with technical support and will mute all participants so we can just hear the speaker and there are no distractions. If everyone's ready, I think we should make a start. It's my pleasure to introduce Professor David Dexter, Deputy Research Director, Parkinson's UK, to tell us about Parkinson's UK research strategy for the next few years. Professor Dexter joined Parkinson's UK as Deputy Research Director, overseeing scientific programmes on the 1st of March 2017. David has 32 years research experience in Parkinson's disease, the last 23 years of which was spent at Imperial College London, where he was Professor of Neuropharmacology and Deputy Head of the Division of Brain Sciences. His research team made significant contributions into understanding the molecular mechanisms that cause Parkinson's and in the development of novel therapeutics. At Parkinson's UK, he manages the University Grants Funding Programme, pioneered the development of a new College of Experts-based peer review system, manages large international projects, and plays a role in developing and running projects with the Parkinson's UK Virtual Biotech Programme. I would now like to hand over to David Dexter.
Thank you, John, and thank you for the research interest group to actually invite me to speak today. I'll start by sharing my screen so I can get my slides up. All right. Okay, hopefully the slides are coming through now. And I'll just get my right. Can everybody see my pointer on the screen as well?
Yes, we can.
All right, great. Okay, so what I'm going to do is give you a brief overview of our strategy. So this was just launched this year and run until 2024. So this really sets out how together we will actually find a cure and also to improve the lives for everybody affected by Parkinson's. So underneath this overarching theme, we've got three main themes. One is getting Parkinson's understood, better support every day, and also accelerating research breakthroughs. But at the center of this is a new initiative called Team Parkinson's, and I'll explain about this on the next slide. So Parkinson's is too big a problem for one charity to solve or one research center to develop an award-winning drug. So what we're trying to do with this new strategy is to generate a powerful movement of people. So everybody can actually make a contribution. whether you're a person with Parkinson's, a supporter, whether you're a nurse or a researcher. So if everybody pulls together under Team Parkinson's, that we can actually drive forward and accelerate drug development and also improve the lives of people with Parkinson's. Now, you may see a little badge, a little blue brain badge in my lapel of my jacket, and that is our emblem for Team Parkinson's. So this is an ambitious initiative, and we're looking to get 150,000 people to join Team Parkinson's within this strategy period. So one of the major problems is that the general public really don't know what Parkinson's is. And this really does pose a big problem because it makes lives of people who are affected by Parkinson's more difficult if the general public don't understand what Parkinson's is. So one of the pillars of the strategy is actually driving up the general knowledge of Parkinson's in the community, and also having a campaign that you may even see us on the television with to improve the awareness. So if we can improve awareness, then what we can do is get people to actually support us and say, Parkinson's has a major effect on people's quality of life. We need to do something about this. Also, we're going to have a campaign with politicians. Our parliamentary group is also going to take a leading role in this as well. So the other pillar to the strategy is better support every day. So the aim of this is really for people affected by Parkinson's and their loved ones is to provide them with the correct support and care that they need for every day within their lives. So whether you're somebody who's newly diagnosed with Parkinson's or has had Parkinson's for a number of years, there'll be a tailored support package for people at every stage. Now, if people will see that in the future, we've got a new initiative coming out. It's been trialed in 5 health centres already, and this is Parkinson's Connect. So this is a service delivery platform, and at the moment, we're connecting with about 81% of people who are diagnosed with Parkinson's, and we're offering support. But obviously what we want to do is get that up to 100%. And we'll be offering personal support over the telephone, tailor-made online information about help and care and extra support that Parkinson's UK and the Excellence Network can actually provide for people whether they're newly diagnosed or they've had Parkinson's for a number of years. So Parkinson's Connect will start from later on this year, be rolled out across the whole of the country. So watch out for this. You can sign up through your Parkinson's nurse or your neurologist into Parkinson's Connect. So obviously what I'm here to do today is really talk more about the research strategy. So the theme for this is actually accelerating breakthroughs in research. So we're developing a pioneering research program that delivers life-changing new treatments for people affected by Parkinson's. And the thing is that we involve people with Parkinson's at every step of this way. So we have people affected by Parkinson's on our research strategy board. We have specially trained advisors who provide lay reviews for our grants programs as well. So people affected by Parkinson's shape what we do in research at every stage. So in the past, we've worked in the area of actually funding research discoveries at the university level. As I'll show you on the next slide, that really what would normally be is, so we'd fund up to this point, and then industry would take over and actually develop new treatments, take them through clinical trials and get them approved so they can be used wider in clinical use. However, industry has pulled away from this area here. So hence in 2017, we initiated the virtual biotech. So also what we've realized is that we don't need necessarily just to keep developing new drugs. We actually need to change the way that clinical trials are run, because at the moment, they're not fit for purpose. So the Critical Path of Parkinson's project, this is a global initiative. So we've got 14 pharmaceutical companies, 6 other Parkinson's charities working with us to change the way that we actually run clinical trials. So, and also, as I mentioned earlier, so we involve people affected by Parkinson's at every step in everything that we do within Parkinson's UK. So this really shows you why we need the virtual biotech. So we would normally fund research at this level, and then the exciting discoveries would be picked up by industry and taken until they're actually on the market, on the shelves, so people can actually use them. But unfortunately, this funding from industry is dried up, so new drugs in development die in what we call this valley of death. So what we're doing is providing this bridging funding to get these exciting discoveries developed into new treatments. So the virtual biotech was set up in 2017 and is new funding from the charity of about £5 to £6 million per year. So what is the virtual biotech? It sounds a little bit strange. You know, we don't have any buildings or labs ourselves. A lot of it is done virtually, and a lot of it is actually done through other companies. So it runs like a regular virtual biotech, biotech company. It creates and tests new treatments, but instead of doing it for profit, we're doing it because we need to get new drugs developed for Parkinson's. And working through this virtual biotech model is one way that we can actually accelerate and drive the drug discovery a lot faster than what we can for necessarily working through universities. So what we've funded so far, we've got 8 virtual biotech projects running so far. So that's about £13 million of investment so far. This year, we've also commissioned 2 new drug trials. So these is cannabidiol. So this is an extract from cannabis, and this is looking at psychosis, which is one of the side effects of common anti-Parkinson's therapies. And also this is on SITraN, which has been in the clinic for a long time as an antiemetic. So these are repurposing type studies that are looking at psychosis and hallucinations in people affected by Parkinson's. Unfortunately, due to COVID, these clinical trials have actually stopped at the moment, but we're looking to actually get these new trials back up and running again. So one piece of news which I'd hope to break with you today is an announcement of a new major trial that we've got co-funded with an international Parkinson's Foundation. But that will be coming to your screens fairly shortly, so watch out for that announcement. But we're assessing about 20 new potential virtual biotechs per year. So what do we actually fund at Parkinson's UK? So we're committed to actually improving the lives of everybody affected by Parkinson's. So this includes people affected by Parkinson's-like conditions like MSA and PSP as well. So our ethos is to drive the development of faster treatments and faster cures. So we're the only charity in the UK that improves treatment, whether it's symptomatic or whether it's developing a drug. And also we are developing a new non-drug approach program that I'll talk about on the other slide. So far to date, we've invested over 100 million in research so far. So people like yourselves affected by Parkinson's helped us develop this list of top 10 priorities. So obviously everybody wants a cure, but next down on the list, we generated this list of priorities. And so our researchers have to focus on these priorities when they're writing applications to us. So also mentioned already that we involve people at every stage. So we are one of the leading charities on public and patient involvement. So we have about 150 specially trained people affected by Parkinson's who assist clinicians in developing their protocols for clinical trials. They also liaise with researchers in developing their research. So this lay input into their programs is very effective. And 99% of researchers have said that the PPI support that's provided by Parkinson's UK has actually been critical in actually shaping their research. And also we've given PPI support to over 200 research programs within the UK. So also we've got lay reviewers that score all of our grants coming into us, and so we're constantly looking for new people to take on this role, and we do have a special training program which allows you to actually do this. So if you're thinking about becoming a lay reviewer of our grants or involved in any other program that we're doing, please get in touch with me at any time. So also, many of you be aware that non-drug therapies, particularly things like exercise programs, can actually deliver benefit to people affected by Parkinson's long before we actually get a new drug on the market. So this year we launched a non-drug therapies award scheme, and this is really to accelerate any potential benefit from non-drug approaches. So these are 2 years in duration, and anybody has to have a clear plan of actually getting that program into the community after that two-year period. So we had a fantastic response from researchers in this first year. We had 23 applications. So we've shortlisted a number, and they've gone on to full application stage. And here you can see a gentleman actually wearing a VR, so this is virtual reality headset. So we've got things like using virtual reality to identify areas that cause freezing in a person with Parkinson's. And so then you can actually develop coping strategies to overcome freezing. So we've also got applications looking at speech therapy. Speech therapy is a very important service, but often access is limited because you need to be in an area where there is a speech therapist. So one of the applications is looking at delivering this through an online mechanism. So later on in the year, we'll be actually making the announcements of the awards for these programs. So also what we're starting to do is actually jump into the driving seat a little bit more. In the past, we've expected researchers to come to us with applications that are going to solve the big problems with Parkinson's. However, now we know a lot more about Parkinson's. We can actually see where the gaps in our knowledge are. So what we've initiated is a new funding stream called Strategic Investments. So here we'll be actually identifying the roadblocks in our understanding, the roadblocks that's stopping us developing an effective drugs. And so they will make some strategic investments in these areas so that we can actually overcome these roadblocks and drive through new drug developments and cures. So also we're partner in international programs. I've already mentioned the Critical Path for Parkinson's project, but we're also IMI partners. So it's the Innovative Medicines Initiative. So this is the largest public-private partnership. This is about 5.3 billion euros. So part of the money comes from industry, like drug companies like Pfizer, UCB, and part of it comes from the EU. And we're involved in 4 of these programs. So one of them is trying to tease out what the role of the mitochondria is in Parkinson's. So one is sharing data across different initiatives. So we're also involved in a trial looking at improving cognition. And the other one is actually looking at digital endpoints to help us understand the effectiveness of drugs in clinical trials. So all in all, we've been able to work with the EU and industry to actually leverage an extra £36.8 million of additional money going into Parkinson's research. So finally, I just wanted to remind people that on the Parkinson's UK website, there is the Take Part Hub. You can just simply put in your postcode in here, and it'll tell you all about the exciting bits of research that you can actually take part in. So You don't have to be taking part in clinical trials. We're often looking for people who want to fill out questionnaires or take part in remote studies. But if you're interested in clinical trials, it will tell you about all the clinical trials that are happening in your area or an area where you're willing to travel to. And then you can actually contact the research teams running those trials. So I'll leave it there, and at the end, I presume we'll be doing the questions through the panel reviews.
Thank you very much, David. It was a wonderful talk that you've given us, covering a wide range of information, and the amount of money that's been devoted to research is very, very impressive. I think Liz may have some questions for you.
Yes, thank you, David, and thanks, John. No questions for David yet in the chat box, but I believe, John, you have some questions that were submitted in advance of the event.
Yes, I have a couple of questions specifically for David. Is Parkinson's UK doing anything to develop the Emma's watch that has been shown to reduce tremor when worn? If so, how is it being taken forward by Parkinson's UK?
Yeah, so probably many of you watched the fantastic program that was done on the watch. So Emma actually now works for Parkinson's UK and is leading on the technology advances that we're helping to improve people's lives. And so this watch, so Emma was a fantastic graphic artist, and then with the tremor, she was unable to perform her usual duties as a graphic artist. So the watch enabled her to actually draw again. So we've been working with IBM on this project, but what we've had difficulty is actually getting them to produce enough of the watches so that we can actually then take them into a clinical study. So what we're trying to do is encourage them to make enough watches so that we can actually do a proper, carefully controlled study to see whether this watch could be something that would be beneficial to control tremor in a wider group of people. So we are still working on it, but unfortunately, IBM are sort of dragging their heels on this a bit.
Thank you very much for that information. In your top 10 Parkinson's priorities for treating Parkinson's, I wondered if they were in priority order, as a recent survey led by Claire Bale in Parkinson's UK found that tremor was the #1 symptom of most concern to people with Parkinson's, and yet tremor is not specifically mentioned on your list.
Yes, the survey that was recently done was at different stages in Parkinson's. So it was looking at people and in blocks of years after diagnosis. And so the importance of different symptoms was actually shown in that study to change with time. And as you mentioned, tremor is important, and it's poorly controlled by drugs at the moment. So it is on our priority list, although that survey, which has got the top 10, was done a number of years ago and was looking more about the non-motor symptoms and balance and falls. But tremor is certainly top of our priority because, as most people know, that if you take your Sinemet or Madapar or take your dopamine agonist, it doesn't have a great impact on tremor.
No, it's one of the most difficult
Yes.
conditions to deal with, isn't it? Most stressing as well.
Yeah.
Well, thank you very much indeed, David, for answering our questions and for your presentation, which has given us a very good overview of the strategic direction that Parkinson's UK wishes to follow.
Thanks.
I'd now like to move on to our next speaker. And Heather Mortiboys, I'm sorry, I'm just trying to share my screen. Can everybody see the screen?
Not yet, John.
Okay. Could you help me with that, Bliss?
You'll have to click on share screen at the bottom of your screen.
Okay.
Do you see that icon?
I'm not seeing it at the moment.
Yeah, if you take your mouse down to the bottom of the screen, your cursor, you'll see it'll pop up at the bottom, share screen in green.
Yes, I think.
I'm not able to do that for you, I'm afraid. There you go.
So my apologies for that. So it's my pleasure to introduce Dr. Heather Mortiboys, who trained in biomedical sciences at the University of Sheffield and continued her postgraduate training on the international PhD programme at the Max Planck Institute for Cell Biology and Genetics in Dresden, Germany. During this time, she investigated the role of mitochondria in neurodegenerative and neurometabolic diseases, with a specific focus on Parkinson's disease. And that links in neatly with David Dexter's indication that mitochondria is one of the Parkinson's UK's research projects. She was awarded her PhD in 2006 with the highest distinction. Shortly afterwards, she moved to the University of Sheffield as a postdoctoral researcher in the Neuroscience department to focus on mitochondria in Parkinson's disease. She is now a Parkinson's UK Senior Research Fellow and Senior Lecturer at the Sheffield Institute for Translational Neuroscience, heading a research group of 10 people, including postdoctoral researchers, PhD students, and research assistants. Heather's research interests and expertise cover mitochondrial components of neurodegenerative diseases, specifically focusing on the role of mitochondrial function, morphology, and DNA in Parkinson's disease. Alongside Heather's research, she is passionate about patient public involvement in research and is a volunteer of a Parkinson's UK Patient and Public Involvement Steering Group, hosts the Yorkshire Research Interest Group, and regularly visits Parkinson's branches, as well as local schools to discuss Parkinson's research. I'd like to hand over to Heather to present her talk.
Thanks very much, John, and thanks very much to you and the rest of the group for inviting me to talk to the group today at this virtual meeting. So hopefully you can see my slides now that I'm sharing with you.
Yes, we can see them, Heather.
Lovely. So John's given me a really nice introduction, and from that, hopefully everybody has gathered that I've been working on Parkinson's pretty much my entire scientific career and really focusing on the mitochondria. So today I'll be talking to you about what the mitochondria are, why I think they're so important in Parkinson's, and some of the research work that we're doing that's funded by Parkinson's UK to look at mitochondria specifically. So the picture on this slide, this beginning slide, is the front of the Sheffield Institute for Translational Neuroscience, or SITraN building. And the large life-size SITraN letters are made with traditional Sheffield steel. And as you can see, it's always blue sky and sunny in Sheffield. And it actually is this morning as well. So John actually has already covered much of this little bit about me section in his introduction. And as he highlighted, I started off life actually in Sheffield doing my undergraduate here. And strangely enough, I now lecture on this BSc in biomedical science course that I once took. So that's quite a turnaround or evolution. And then I moved to Germany, right to the eastern part of Germany in Dresden, to do my PhD at the Max Planck Institute there, studying mitochondria and their role in various conditions. Stayed in Germany for a little bit longer and then moved back to the UK, really focusing on mitochondria in Parkinson's. And I've been heading up my own lab at SITraN since 2013, when I was awarded the Parkinson's UK Senior Research Fellowship. And as John said, the team now is actually increased quite substantially over the recent months, and we're about at 15 to 17 people focusing on metabolism in neurodegeneration. So both John and I have mentioned mitochondria a lot, but what are they? So they're essentially the batteries of all of our cells, and they generate most of the cell's energy. And that energy currency in the cell is called ATP. So they're in all of our cells, every single different type of them, a heart cell, skin cell, different types of brain cells, all contain several different mitochondria. And the mitochondria move around those cells to get to the areas that really have a high energy demand. And actually the brain cells are some of the cells in the body that have the highest demand for energy. So just like us as a whole body, the mitochondria can use different types of food to make that energy. So they can use different sugars, different fats to make energy. Some very efficiently, and they can make lots of energy from some sugars going in, and other types of food going in, actually they can make energy, but they do it less efficiently. And the other thing to note about them is that I've drawn them on this little diagram almost as little ovals, but actually they're extremely dynamic in their shape. So they can change their shape depending on the energy demands or the cell type that they're in. And so some shapes of mitochondria will make energy more efficiently than other shapes. So this is just some skin cells that we've grown in the laboratory, a single skin cell, and we've stained in blue the nucleus of the cell, so that's the organizing center of the cell, and we've stained in green the mitochondria. And so hopefully you can see that this cell at the top here, the mitochondria, have all interconnected and they've formed a very elongated network. Whereas the cell at the bottom, the mitochondria are more fragmented, they're more broken up, there's more individual, much smaller and shorter mitochondria. And so this is quite important for how efficiently those mitochondria can actually generate energy. So why are we interested in mitochondria in Parkinson's? So actually the first hint that there was something potentially wrong with the mitochondria in Parkinson's came in the 1980s, when in California there were a group of drug addicts who came down with something very similar to Parkinson's clinically. And it was discovered that the reason why these people had developed the Parkinsonism was because the heroin they had taken was contaminated, and the substance with which it was contaminated, it was found, went straight to the mitochondria, and once there, stopped the mitochondria from working. So after that, and for the subsequent decades, there's been much research into mitochondria in Parkinson's. So people that have looked at post-mortem brain samples of people with Parkinson's, again showing that the mitochondria were not working so well in the post-mortem brain samples from people with Parkinson's. And people have also looked then at peripheral tissues, skin cells, muscle cells and blood cells. And although there's variability, pretty much every study has found that there is something wrong with the way that the mitochondria are working, even from those peripheral cells from people with Parkinson's. In addition to that, then the genetic animal models have also been generated, whether it be a fruit fly model, a zebrafish model, or a mouse model, and they have also shown that the mitochondria are not functioning as well in those genetic models of Parkinson's. So what do we do in the lab? What does my group focus on doing? So we really focus on using samples taken from people with and without Parkinson's. And so one of the ways that which we can do this is we can take skin biopsies from people. And if this was a live in-person event, I would show you the scar I have on my forearm from where I too have had a skin biopsy taken. So it leaves a very, very small scar, and we can set up these skin cells and we can grow them in the laboratory. The picture down here on the right is a picture of some of those skin cells that we can grow in the laboratory. And then what we can do is we can look at the size and the shape of the mitochondria in those skin cells, like the pictures I showed you earlier, and we can also look at then how well the mitochondria are functioning, how much energy they're producing, and some of their other functions. So what we have found over a number of years of work, and what's also been found by many other researchers in the Parkinson's research community, we have found that the skin cells from people with Parkinson's have lower levels of ATP, this energy currency in the cells. So the mitochondria are producing less energy. We have also found that the mitochondria have changed their shape. So they're not the same shape as the shape of people without Parkinson's. So what we did was, once we investigated this in the skin cells from people with Parkinson's, we wanted to say, okay, well, can we treat those skin cells with a drug and that can boost the energy levels in those cells? And so we looked at a variety of different drugs, and actually we found several drugs which could increase the energy levels in the skin cells from people with Parkinson's. So I just want to talk a little bit about drug screening in Parkinson's. So David very nicely has introduced the Valley of Death region that the Parkinson's UK Virtual Biotech is funding, that area. But if you think about drug screening more generally, then you've got several phases. A discovery phase, where you will be screening thousands and thousands of different experimental compounds in some kind of a cell model system, perhaps in the laboratory. We'd then go through an optimization phase of really finding the right compound that you could take forward as a drug candidate. Once you had that drug candidate, there's a safety phase, then a full preclinical phase, really assessing everything else that could possibly be known about that drug candidate. And then you would get into the phase 1, 2, and 3 clinical trials. And this process conservatively can take 10 to 20 years. And as you can imagine, the costs for the whole process are really quite large. I've put on here approximately £800 million. Some people would also estimate up to £1 billion. So what we wanted to do was we wanted to take that slightly alternative approach and not start with completely novel compounds, but use a drug repurposing approach. And David touched on it in his talk, that this is where we can take a drug which has already been through that entire process and been found to be effective for use in one condition, and see whether it can be effective for people in Parkinson's. So this, in fact, was the project that was funded by Parkinson's UK for my senior research fellowship. And so what we did in that project was we grew the skin cells from the people with Parkinson's, and we put them into 384 well plates, which are shown here in this picture. So we put a couple of thousand cells into each of those wells. We then used state-of-the-art machinery that really isn't widely available in an academic university environment, and is more at home in an industry environment. And it uses a little bit of science fiction, I think, to shoot the drugs onto the cells in this 384 well plate. And then we leave them for a little bit. We put in a variety of fluorescent dyes, which enables us to take pictures, and then we can generate thousands upon thousands of images of the mitochondria from the skin cells of people with and without Parkinson's, with and without treatment with these different drugs. So this is a picture of Hannah, who worked in the team with me doing this screen. So she's working in the tissue culture cabinet. She's culturing the skin cells from the people with Parkinson's and putting them into those 384 well plates. And we use skin cells from 3 different groups of Parkinson's people. We use them from people that had a known early onset genetic cause, from people that had a known late onset genetic cause. And then we also use them from people with the largest group of people with Parkinson's, so-called sporadic Parkinson's, where actually we don't know why those people have developed Parkinson's. So Hannah plated those skin cells into the 384 plates. We went through that entire screening process. We generated more than 2.5 million images of the skin cells from those people, and then we set about the rather large task of analysing all of those images. And the actual really exciting bit of the study is getting to the results. So what did we find? So we found 29 compounds that had a very, very positive boosting effect at boosting the activity and the function of the mitochondria, enabling them to produce more energy in those skin cells from people with Parkinson's. And really excitingly, actually, we found that several of those compounds worked in all 3 groups of cells. So those from the people with an early onset genetic cause, a late onset genetic cause, and from the largest group of people with Parkinson's, those with sporadic Parkinson's. We did find a couple of compounds that preferentially worked on only 1 type of Parkinson's, but actually a lot of them that worked across all 3. So that's all in skin cells. And you're probably all sitting there thinking, well, hold on a minute, Parkinson's is not a condition that predominantly affects the skin, it's the brain cells which are prominently affected. And you're absolutely right. But we can't take very easily brain biopsies from living people. So is there a way in which we can study the type of brain cells affected in Parkinson's? And there was a model revolution that happened a few years ago now that Yamanaka got the Nobel Prize for. And this model revolution enabled us to change the skin cells and fool them and change them and turn them into a stem cell. And a stem cell is a type of cell that then can go on to become any type of cell in the body. So we could then use this technique to use those skin cells that we had been using all along, but turn them into the stem cells and then into the specific type of brain cells affected in Parkinson's. Now, one of my colleagues at Sheffield, shown here, Laura Ferrulio, actually modified that Yamanaka technique a little bit. Because what happened during the Yamanaka technique was the reprogramming reset the cell back to how the cell would have been at an embryonic level. So all of those changes that have happened in the cell to the DNA and other parts of the cell over a person's lifetime as people age, they were lost with that other reprogramming technique. And as you know, age is one of the biggest risk factors for developing Parkinson's. And so we thought it was really crucial that we try and keep the age changes that have happened in people's cells throughout their life. And the little tweak to the protocol that Laura developed actually enables us to do that. And so Laura tweaked it a little bit to go to direct reprogramming, which means that we can go from skin cell to a type of stem cell that can only become different types of brain cells. And so we couldn't make heart cells or liver cells from those type of stem cells, but we can make the dopaminergic neurons and also the astrocytes and other supporting cells which are affected in Parkinson's. So we did this direct reprogramming technique with all of our 5 skin cells that we have from the people with Parkinson's that we used to generate that data I showed you. And then we tested those drugs. And so we said, did those drugs actually work on the skin cells from, on the brain cells, sorry, from people with Parkinson's? Did they have an effect at boosting that mitochondrial function? And the short answer is that some of them did, yes. So great. We've taken a bunch of compounds which are already in clinical use. We've looked at them across lots of different skin cells from people with Parkinson's. We've found some that boost the energy status, the mitochondrial function. We've then turned those skin cells into dopaminergic brain cells. We've looked, some of the compounds are still active and still boost the mitochondrial function in those dopaminergic brain cells. So what are we doing now? What are the next steps? So that kind of brought to the end the funding of my senior fellowship. And then I developed with the Parkinson's UK Virtual Biotech team a funding application which actually took the top 2 compounds from that original study and made some chemical tweaks to them. Because what we found was that those top 2 compounds, although they were already in clinical use, actually they had some long-term side effects that would not be good for people with Parkinson's. So we wanted to see whether we could chemically modify them to get rid of those long-term side effects that would not be good for someone with Parkinson's, but keep the mitochondrial boosting effects. So this project was funded from the Parkinson's UK Virtual Biotech, and that is now coming to a close, slightly delayed with the COVID situation, but that project is the first batch of streaming is coming to the close, and I'm working very closely with the Virtual Biotech team to put together subsequent project planning to take those compounds forward. So just to summarize, we found that there's a fundamental problem with the mitochondria in cells from people with Parkinson's. We went about testing thousands of different drugs to find a drug which restores healthy mitochondria. Some of those best drugs, however, were found to have side effects that would not be good for someone with Parkinson's. So we've now used chemical modifications to design new compounds, new drugs to minimize those side effects, but keep the mitochondrial boosting effects. And we have, we're testing those in the laboratory now, but we have found compounds that we can do that, and we're therefore developing the next stage plans along with the Parkinson's Virtual Biotech team. So finally, I just need to thank my team. So this is a slightly out of date photo, I have to admit, of my team on the balcony from SITraN, and I've put a big red arrow for Tom and Aurelie, who undertook really quite a lot of the work, or most of the work that I've spoken to you today about. As with, I would think, most scientific projects now, it's not just the team in Sheffield working in isolation. We work together with lots of other scientists around the UK and around the world to really develop the science as quickly as possible. In particular, a chemistry associate professor at the University of Sydney in Australia, and a clinical professor, Jan Aisley, in Norway. And the final slide I will just put up very briefly is that earlier this year, SITraN won the Queen's Anniversary Prize for Innovation in Neuroscience, and that was specifically for work that we had done involving people affected by neurodegenerative conditions such as Parkinson's, and developing progress towards new treatments for them. And I was lucky enough to be one of only 5 people from SITraN that got to attend the event at Buckingham Palace. And if you can see, this is me here, and I'm even wearing the Parkinson's UK turquoise blue colours on my dress. So with that, I'll finish, and hopefully, John, I managed to stick to time.
Thank you very much indeed, Heather. That was a wonderful talk, and I think you have kept to time very well indeed. It was great to hear about the progress that's been made and the possible new drugs that hopefully will provide much better treatment for people with Parkinson's in the future. And wonderful too that you got such a wonderful award for your university.
Thanks, John. I have a couple questions here in the chat box, if that's all right.
That's fine, Liz. Thank you.
So the first one is from Helen, and Helen asks, the mitochondria change that you see, is it best described as a cause of Parkinson's or more a result of Parkinson's?
So that's a really key fundamental question. And so we're also working on that question to really understand. So where our understanding is at the moment is that there is something wrong with the mitochondria pretty much in most people with Parkinson's. And the research would suggest at the moment, so we had a big study that was published earlier this year, also funded actually by Parkinson's UK, that indicated that perhaps approximately 1/3 of people with so-called sporadic Parkinson's would have a mitochondrial driver really causing their Parkinson's. And the other 2/3 of people would have a different cellular component perhaps driving their Parkinson's, so really being the cause of their Parkinson's. But downstream to that, they would also have things going wrong with their mitochondria. So with a mitochondrial targeted therapeutic, we hypothesize, or we think, that it will have the most effect for that approximately 1/3 of people with the mitochondria really is the driver or the cause of their Parkinson's, but it will still be effective to the rest of the people with Parkinson's because downstream the mitochondria are still having issues in those people as well.
Thanks, Heather. And a second question from Anil. How long do we think it will be before we see new drugs available that will be used to boost mitochondria? Are you able to give us any sort of idea of time scale?
Yep, so drug screening and drug discovery research is very much an iterative process. And so the first drug that we actually found in the skin cells from people with Parkinson's that could boost the mitochondria was a drug that's called ursodeoxycholic acid, or UDCA. And that actually was already in clinical use for people with various liver conditions. And so a colleague of mine at Sheffield, Professor Oliver Bandmann, has taken UDCA already into clinical trials in people with Parkinson's. And in fact, that study is ongoing still, even with the slight variations that have been needed due to COVID. But those people have already been recruited and along taking UDCA. There's also another study looking at UDCA in people with Parkinson's being run by some clinicians in Minnesota and in the USA as well. And so there are a variety of mitochondrially targeted or boosting therapies which are at various stages of the drug discovery pipeline. Some of them already in clinical trials, some of them, let's say, a couple of years away, or maybe only a year away from clinical trials, and some of them then much earlier. But we're learning every single time we go through that process. And it's not that we don't think UDCA will be good. We do think UDCA will be good, but we're still then trying to go back and find something that we think might be even better.
Thanks, Heather. And another question about these drugs in development, when they are, or hopefully when they become available, will people who have gone through deep brain stimulation surgery, DBS, be able to take them as well?
So that will depend very much upon the drug itself. So it might be different for different mitochondrial boosting drugs. I know that generally for setting up of clinical trials, then people who have had DBS are not included in many clinical trials, but that does depend on the clinical trial makeup. And I think some of the work that David highlighted earlier with understanding much more about the various readouts that we can use in a clinical trial for a drug which may have neuroprotective properties will be helpful in also then being able to maybe widen the Parkinson's community which can take part in trials.
Thanks, Heather. That question was from Jan, and I have a question from Jane. Is it possible to tell if your Parkinson's has a mitochondrial driver?
So for people with Parkinson's at home, if you like, probably not. That's probably quite difficult to tell. So one way in which we are studying that, and actually on the back of our study that was published earlier this year, I'm interviewing next week for a PhD student to specifically really investigate this. The way that we're approaching it is looking at skin cells from tens, if not hundreds of people with Parkinson's, and also then looking at the mitochondria, but also the other main pathways involved in Parkinson's, and trying to really then understand if this approximate third of people with mitochondria being the driver is the real sense. So in the laboratory and with those studies, that's how we're approaching it. But for people at home with Parkinson's, I don't think really there's an easy way to be able to identify that.
Thank you. Do I have time for one more question, John, or is it time to move on?
We have time for one more question, Liz.
All right. thank you. The last question is from Helen. This might be for all of our speakers, perhaps. Helen comments she wasn't sure you could differentiate between Parkinson's with a genetic cause or an idiopathic or sporadic cause. So she's keen to know a bit more about how you might identify people that have a genetic link to their Parkinson's.
So maybe I can answer first, and then if David or Simon have anything to add, then they can add to that. So when in the talk, when I referred to people with a genetic cause of their Parkinson's, that's where they have had genetic testing, and there has been a change in one of their genes identified, which is known to actually cause Parkinson's. And so some of the genes, for example, Parkin or LRRK2, LRRK2, that you may have heard other people talking about. And that genetic testing is provided then on a clinical service level. I know access to that genetic testing can vary depending where you are. The other part of genetic risk for Parkinson's is genetic changes which increase your risk of developing Parkinson's, but are not definitely 100% causative for Parkinson's. And it is thought that in that group of idiopathic or sporadic people with Parkinson's, actually very many of them will have genetic changes which increase their risk, but are not 100% causative. And so that's how we separate out the genetic link to Parkinson's as being 100% causative, and the sporadic people with Parkinson's who may have an increased risk based upon their genetics.
Thank you, Heather. I think that's all of the questions we have at the moment, John.
Thank you. I just have one question. I'm not sure if it's been covered. May I ask that, Heather? Do you advocate people with Parkinson's taking terazosin, SITraN in brackets, recently shown to slow Parkinson's progression? And if so, at what dose?
So I think the data around terazosin is actually really interesting. So the mechanism, if you like, of that having an effect is around switching how the cell can produce the energy, so how it can maintain its energy levels. So actually switching it away from a mitochondrial pathway, switching it to other cellular pathways which can maintain the energy levels. In terms of if I would advocate people taking it and at what dose, then I would advocate people taking it until it has been fully gone through the clinical trials for Parkinson's and fully tested in that level. And I have to say, I will have to defer to David or Simon as to exactly where those clinical trials and what stages they're at.
Yeah, I think the trials have been initiated, I think, in the States. And also, it's not widely used as a drug for prostate problems in the UK. So both the Tracking Parkinson's cohort and the Oxford cohort have looked to see how many people in the cohort have actually taken terazosin, and it's actually quite small because unlike in the States, they're using a different drug to control prostate problems. But if you're taking a current therapy for your prostate, you could also talk to your clinician, as some people are doing, is changing to terazosin as the form of treatment for it.
Thank you, David. That's very, very helpful. Did you have anything to add, Simon?
Only that we have to be very careful addressing these sorts of questions, as terazosin can also have side effects with regards to blood pressure, etc. So these sorts of questions should really be addressed to a neurologist or your clinician, as opposed to online forums like this.
Yes, it's fully understood. Thank you for those comments, Simon and David. And Heather, thank you very much indeed for such a wonderful talk on the mitochondria. and all the research that you and your team are doing. I was particularly impressed with the creative way that your colleague has improved the cost effectiveness of the research. Just amazing.
Thanks, John. Thank you.
Well, now it's time for us to have a short break for about 10 minutes. And I suggest we come back at 20 to 12 when Simon Stott will be giving us his presentation. Look forward to seeing everybody back then. Thank you.
I think we're back from break, John, and ready as soon as you're ready.
Okay, thank you. Welcome back, everybody. Hope you had a good break. Now, time for coffee. And I'm just getting ready to share my screen. So, hope everybody's raring to go again. Welcome back, everybody. I'd like to introduce Dr. Simon Stott, who is the Deputy Director of Research at the Cure Parkinson's Trust. The Trust is an international supporter of both lab and clinic-based research on Parkinson's. It currently has 16 compounds being evaluated in 17 ongoing clinical trials for disease modification in Parkinson's. And the Trust is hoping to increase that number to 25 trials over the next 18 months. Originally from New Zealand, Simon has over 15 years of experience in the field of Parkinson's research in both the academic and biotech sectors. He has been involved in lab-based research as well as clinical studies with numerous scientific publications. During his career, he has authored or co-authored more than 25 scientific publications, has been involved in lab-based research as well as clinical studies, and his associations with world-class research groups has provided him with a wide international network of connections. Simon also takes a very active role in patient engagement, regularly speaking to Parkinson's support groups and helping to raise awareness of the condition. The science of Parkinson's is Simon's personal perspective of what is happening in the world of Parkinson's research, with regular updates explaining the science behind the new discoveries, with up-to-date clinical trial results, and introducing some of the people behind the research. I would now like to hand over to Simon.
Thank you, John. I'm not sure how to live up to that introduction. I'm going to share my screen now, and we can get started. There we go. So the title of my talk today is Monsters, Mucus and More, and it's an overview. It's a very small overview, a brief glimpse at what is going to be happening, what is happening right now in 2020, but also what's going to be happening in 2021 with regards to Parkinson's clinical research. And I'm primarily focused on disease-modifying therapies, so therapies that are going to slow, stop, or reverse Parkinson's. And that's partly because I work for this charity called the Cure Parkinson's Trust, where we UK registered, but we fund internationally research that's focused on disease modification for Parkinson's. It was begun by these 4 gentlemen here, and I should add their extremely supportive wives. In 2005, they were frustrated by the lack of research that was going into curing the condition. They didn't want new symptomatic therapies. They wanted something that's going to cure and get rid of this condition. So they started this charity. And one of the principal individuals in this effort was this gentleman here, Tom Isaacs. Tom was diagnosed with Parkinson's at age 27. And he naturally did what any normal human being would do when diagnosed and given this devastating news. He decided to walk the entire coastline, all 4,500 miles of England. And it took him a year. And after that year, he came back and he set up the Cure Parkinson's Trust, which is now obviously a major funder of Parkinson's research focused on this idea of disease modification. We do both preclinical as well as clinical research. And our clinical research is primarily done through this program called the Link Clinical Trials Initiative. That is where every year we have a group of 20 to 25 of the world's leading experts on Parkinson's, and also a large group of Parkinson's research advocates gathered together for a 2-day meeting. And they evaluate 20 to 25 compounds. We make dossiers, large books of all the information that we know about these compounds. And they all of them must have some evidence of disease modification or potential for disease modification in Parkinson's. And this committee's job is to prioritize which one they think should be going through to clinical evaluation in the next 12 months. And once those agents are prioritized, that's where CPT is mandated to actually take them and get them into clinical trials. But it's not just the committee that is playing an important role here. We have representatives from all the major stakeholders also in the room, from government organizations like NIH and NINDS, to groups like Michael J. Fox, Parkinson's UK, Shake It Up Australia, Parkinson's Foundation. They're all there in the room, and everybody's got a voice and allowed to have an opinion with regards to each of these compounds. Now, what do we mean by disease modification? This is an important concept. The typical trajectory with Parkinson's is that it's a progressive condition. Over time, people are gradually going to get worse. And the typical trajectory is that you have some sort of indication that something's not quite right. It might be a twitchy finger or toe, and then gradually this becomes more of a tremor and there's some rigidity kicking in, and then you'll get a diagnosis from a neurologist. And at that point, you will quite often be put onto what's called a symptomatic treatment. Now, this is a treatment that will cover up the symptoms of the condition, but underneath the surface, the condition is still progressively getting worse. And so a disease-modifying therapy is any medical treatment of a disease that can change that trajectory of that course. It's a therapy that can slow, stop, or reverse the disease. So what the goal is here is to alter this trajectory, this blue line that's on a gradual sort of downward trajectory. If we can gradually sort of lift it up after that point of diagnosis, ideally before the point of diagnosis as well. But at the moment, just trying to raise that trajectory and improve quality of life for individuals with this condition. And at this point, what I'd like to do is point you towards a recent overview of clinical trials for Parkinson's. It was during the last 12 months of 2019 to 2020. And this work has been really sort of led by these 3 Parkinson's research advocates, Sue Buff, Gary Rafaloff, and Kevin McFarthing, where they analyzed all of the clinical trials. They took all the clinical trial data that's currently on a website called clinicaltrials.gov, and they analyzed all of the trials that have got Parkinson's as their main indication, their main disease of interest. And they've broken it down and analyzed exactly what's being done in what areas with regards to symptomatic treatments and also disease-modifying therapies. And here is a large circular map of everything that's going on from phase 1 to phase 2 to phase 3. Phase 1 is just basically a safety test of a drug. Does it kill anyone, basically? And then you go into phase 2, and that's where it's a safety test. You're testing the drug in your population of interest. So does it kill people with Parkinson's? No, it doesn't. Okay, that's good. But there's also, you're looking for evidence of efficacy, some signals of efficacy. Can this drug actually do what it's supposed to do in your population of interest? And then in phase 3, this is a much larger, much longer study, evaluating, again, long-term safety of the drug, first and foremost, but also does it actually do what it does, what it's supposed to do with regards to helping people with these medical conditions. And what Sue, Gary, and Kevin found was that currently, well, during 2019, there were 145 clinical trials for Parkinson's. And pleasingly, 57 of those, so almost 40%, were focused on disease modification. So all of the drugs you can see labeled across the top of the circle here, these are all focused on disease modifying efforts. And the concerning part, when you look at this, is that if you look at the symptomatic treatments in the circle below in phase 3, there's lots of symptomatic treatments coming through. But for disease modifying treatments at phase 3 level, there's only 3 drugs being tested. And so what we really need to do is increase this number of drugs being tested at the phase 3 level. This is the crucial level of clinical testing. This is the step before the regulators say yes or no to a drug being approved or not for clinical use. And so that's really the goal here, is to increase the number of phase 3 trials being conducted. From the standpoint of the Cure Parkinson's Trust, we were very pleased to note that over 10% of the disease-modifying trials are actually being supported by us. And what I'd like to do now is to discuss a few examples of these trials for disease-modifying therapies, or potentially disease-modifying therapies. And the first one is where the title of my talk, Monsters, comes in. So this is going to be about a class of drugs called GLP-1 agonists. And these are drugs that are used for diabetes. They are used by folks with diabetes to control the level of glucose in the blood. And the monster part comes in because the molecule, it is exenatide, came from the venom of this cute little guy. This is a Gila monster. They live in North America, and the bite is actually poisonous. So was that a question? No questions? Okay.
Sorry, Simon, I just muted someone. It's some background noise. Apologies.
No, that's okay. But they might be taking objection to the picture or something. I don't want to offend anybody. So this guy here is a Gila monster. He spends most of his time underground. But when he does come out, if he bites you, it's poisonous. But in that poison, there is this molecule that scientists have turned into this drug called exenatide, and it's used by people with diabetes. Now, back in 2008 or 2009, I think it was, some folks noticed that exenatide also has other properties in addition to modulating the levels of blood sugar. And they found that it actually has neuroprotective properties in preclinical models of neurological conditions. And so they started experimenting in models of Parkinson's, and they found that there too, exenatide was having beneficial effects. And this led to a clinical trial, which was initiated by this chap here. This is Professor Tom Fultonay from UCL, University of College of London in London. He conducted 2 studies. Firstly, one to check that the drug is safe in people with Parkinson's. But then in 2017, they published the results of the second study. And the second study was conducted in a blinded fashion. So that means that neither the investigators nor the people taking the drug knew whether they were taking exenatide or if they were taking a placebo treatment. And what Professor Fultonay and his team found was that the people who are on the placebo drug, they're the red line here in this graph, they progressively got worse over the course of the study, but the people on the exenatide treatment better and were stable across all of the 48 weeks of the study. And then when they removed exenatide, it was only then that they started to show this trend back towards the baseline level. So this was a very exciting result. And I should point out this result was only in specifically the motor features of the condition of Parkinson's. Exenatide in this study only worked for the motor symptoms. And you can have a look at the study via this link. Don't bother writing all this down, by the way. I'm happy for these slides to be made available for anyone who asks. Just more recently, in fact, literally a week ago, there was this study published, again from Professor Fultonay's team, where they went and they had a look at people with diabetes, and they looked at medical records from a large UK database. And they said, what happens to these people who are taking, who have diabetes and are taking this drug called exenatide over time? What does it increase or decrease their risk of developing Parkinson's? And what they found was that the folks who are taking exenatide or GLP-1 agonists like exenatide had a reduced risk of developing Parkinson's. So this was further support for justifying the testing of this therapy in people with Parkinson's, and that is now ongoing at the phase 3 level. So this is the critical test, you could say, before the regulators say yes or no to this treatment being used in the clinic for people with Parkinson's. And so this phase 3 study is now recruiting, and they're looking to recruit 200 people with Parkinson's, and they will be taking the drug or exenatide or placebo for 2 years and being assessed over that time. So we won't see the results of this study until about 2023, 2024, and more likely 2024, I should say. But what's really exciting in this field is that this is not the only GLP-1 agonist that's being tested. Liraglutide is another exenatide-like drug, and that's currently being tested in a phase 2 study in Los Angeles. These people specifically have Parkinson's and are insulin resistant. So they are borderline, if not diabetic, they are borderline diabetic. And we should have the results of this study in 2021. And there's also elixazenatide, another GLP-1 agonist study, which is currently being conducted in France, and that's recruiting 158 people, and they'll be treated, well, they are being treated over 12 months, and the results of that are also expected in 2021. So this is something to look out for in 2021. In addition to those, what's really quite fascinating about this whole exenatide story is that this tiny little charity called the Cure Parkinson's Trust initiated a couple of clinical trials with support from folks like Michael J. Fox Foundation, etc. But this has now encouraged other parties to get into this field as well. And you're seeing large pharmaceutical companies like Novo Nordisk, and they have a phase 2 clinical trial currently going on in Norway. There's another exenatide phase 2 study being conducted in Sweden, and a biotech company called Neuroscience. They've got a new version of a GLP-1 agonist specifically designed for neurological conditions. And that company currently has a phase 2 clinical trial for people with Parkinson's. So what you want from great science is for it to raise lots of questions and generate lots of excitement and activity. And hopefully the exenatide story is going to be one of those classical cases for history. Moving on now, I'm going to move on to mucus, another part of my title. And this involves a drug called ambroxol. So ambroxol is a medication that's used for respiratory conditions. It reduces inflammation in your throat, in the esophagus, and also clears up mucus. But a few years back, some researchers found that one of the other effects of ambroxol is that it boosts the levels and activity of a molecule in the body called GCase, glucocerebrosidase. And this enzyme plays an important role in the waste disposal system of cells of our body. And so the idea here is that if we boost the level of this enzyme and its activity, we will hopefully make cells healthier and they can perform better. In addition, the interesting aspect of this particular enzyme is that people with a very specific set of genetic mutations, genetic variants in this gene, in the gene that produces GCase, have a higher risk of developing Parkinson's. So this led to a lot of preclinical research focused on testing ambroxol in models of Parkinson's and models of this GBA-associated Parkinson's. And then that led to this chap here, Professor Tony Shapira, also at UCL, starting a phase 2 clinical trial for ambroxol called AIM-PD. And that followed 18 people for 6 months who were taking, this is incorrect actually, all of them were taking ambroxol. And these people are absolutely heroic because it required them to take 21 pills a day, and the pills have a very, very bitter taste. So on top of their Parkinson's medication, they were also taking 21 bad tasting pills per day. An absolutely heroic effort. But what was really, really interesting was that overall the drug was well tolerated in these individuals over 6 months. And it also raised the levels of GCase in the brain. As you can see here, the baseline levels are all these little dots, these white dots and black dots. And across the entire group, it was significantly raised after 6 months. And the next stage of this clinical program is now in development. And we should hopefully hear more about that soon. If you are interested in potentially getting involved with that ambroxol research, I'd point you towards this website here called PD Frontline, which is trying to recruit cohort-ready, clinical trial-ready cohorts for these kind of trials. Again, I'll make these slides available so you can follow up if you are interested. Moving on now to the and more part of my title for my talk today, I wanted to make a comment with regards to immunotherapy. So immunotherapy is a way of boosting the immune system of the body. Our bodies produce these Y-shaped proteins you can see here. This is an antibody, and antibodies are used to label entities in our body that the body wants to get rid of, and the immune system comes along and uses that antibody as a bit of a red flag and grabs whatever the red flag is bound to and gets rid of it, chews it up and spits it out. Now what researchers have done is they've used these antibodies, they've designed specific antibodies to target proteins that are associated with Parkinson's, and one of those proteins is called alpha-synuclein. It sounds like a distant galaxy, but it's one of the most common proteins in our body, in our brains. It makes up about 1% of what goes into a neuron. And in Parkinson's, what we see is alpha-synuclein starts to cluster together and form dense aggregates. And it also appears to be being passed between cells. And so with immunotherapy, what the researchers are hoping to do is to grab the alpha-synuclein while it's being passed between cells and get rid of it, and in that way slow down the progression of Parkinson's. And recently we've seen a large clinical trial called the Pasadena trial. So this is a phase 2 clinical trial involving 300 people that was conducted in the US by the pharmaceutical company Roche and Prothena over 52 weeks. And in April, the two companies announced that their immunotherapy treatment had not reached its primary endpoint. So it had not achieved its primary goal for the clinical trial, but they did see signs of efficacy. And so everybody was kind of a bit confused as to how to take this news. Is this bad news or is this good news? Then just recently, last month, they presented some of the data. And what you can see here is that this is the primary endpoint of the study. So the main goal of the study was to see a difference between the blue line here and the red line with regards to the Movement Disorder Society's UPDRS. This is the Unified Parkinson's Disease Rating Scale. If you go in to see your clinician, they'll quite often use the UPDRS to assess your Parkinson's. And the study was focused around part 1, 2, and 3 of the UPDRS. So these are 3 components that are pulled together to give you a score. And when you look at the blue line and the red line, you don't see much difference between the placebo group, which is the blue line, and the pooled treatment group. So 2 doses of the treatment that were given. So you don't see very much difference between these 2 groups. But when you break down the results and you just look at the motor features part of the UPDRS, so part 3 of the UPDRS looks just at the motor symptoms of Parkinson's, and it's clinician assessed, that's when all of a sudden they saw this separation between the red and the blue line. The blue line had a higher rate of progression as opposed to the reduced rate of progression in the red line. And they also they've put a green circle around this plateau part towards the end of the study, where they're suggesting that maybe the progression actually slowed down to a point of not progressing at all. These should be considered preliminary results. This is a phase 2 study, but it's the phase 2. The point of a phase 2 study is to give us evidence or signs of efficacy and whether it's worth continuing with this treatment. And in this particular immunotherapy case, Roche and Prothena are proceeding. They're very encouraged by this result. And so it'll be interesting to see what happens in 2021 with regards to where this program goes next. And they're not the only ones with an immunotherapy approach. The pharmaceutical company Biogen also has a immunotherapy approach in phase 2 clinical trials at the moment. We won't hear the results of that study until the end of 2022 though. But we will see the start of a new form of immunotherapy. This is a vaccine for Parkinson's. It's being developed by a company called Aferis, a company in Austria. They've been conducting phase 1 studies for 4 years now, and they've seen no problems with regards to safety. And so they're shifting to phase 2 later this year. Well, there's not much left of 2020 now, so maybe in 2021 we'll see the clinical trials start. But this is something else to look out for in 2021. Mitochondria, here's outlined the wonderful research that they've been doing up in Sheffield, and I just wanted to highlight the study that they're conducting there as well. This is with regards to UDCA. This is a bile acid that's used to break down gallstones and treat liver conditions. It's clinically available, but Heather and her team in Sheffield have demonstrated that UDCA exerts beneficial effects with regards to mitochondrial activity, and that led to the initiation of the UP study, or UDCA in Parkinson's. It's a phase 2 clinical trial that's recruited 30 individuals with Parkinson's, and we should hopefully be seeing the results of that study in 2021. That should read 2021. Sorry, typo there. Another thing to watch out for in 2021 is the Australian Parkinson's Mission. This is a large clinical program focused on Parkinson's, disease modification in Parkinson's. that's been funded by the Australian government to the tune of $30 million. The difference here, though, is that they are taking multiple treatments in one study and comparing them to a placebo. So there's different groups of people with Parkinson's being treated with different experimental treatments, and they're being compared to one group that's being treated with placebo. The idea here is that you can get 3 or 4 drugs tested at one time, as opposed to just one drug being compared with placebo. Excuse me. And the first of these studies has now started. The first of the APM trials have started. And the second multi-arm study is now in planning. Something else to look out for is the SIMPROVE clinical trial. Excuse me, I'm going dry here. This is a pilot study evaluating a probiotic in 70 participants with Parkinson's. Half of them will be getting the SIMPROVE product and the other half will be getting a placebo for 3 months. And there's been a lot of data recently suggesting that there are issues in the gut of people with Parkinson's, and whether this is an interesting study evaluating whether we can modulate that activity in the gut, and if that will have a beneficial effect on the progression of Parkinson's or the symptoms of Parkinson's. We shall see. 2 more drugs before I finish up. Nortriptyline, this is an antidepressant drug that's being repurposed for Parkinson's. It's demonstrated beneficial effects in models of Parkinson's, and is now about to start a clinical trial called ADEPT PD. This is going to be across the UK. It's another trial to look out for. And SITraN, this is an anti-inflammatory drug that's used for conditions like rheumatoid arthritis. Inflammation seems to be playing an influential role within Parkinson's. And this clinical trial will be starting in Cambridge here in the UK as soon as COVID allows. And they'll be looking to recruit 60 participants for a 12-month period of evaluation on this drug, or placebo, excuse me. Now, one question, as I finish up here, one question I often get is, hey, Simon, what do you think is going to work? We've got all this exciting research going on at the moment. What do you think, what would you, if you were a betting man, what would you put your money on? Now, I've got 2 answers to this question, and before I give them to you, I have to warn you that one of them is going to upset you, but I will explain myself. The first answer is that it depends on the type of Parkinson's each individual has. For example, with exenatide, it is apparent that not everybody is responding to, everybody in that study, the phase 2 study, not everybody in that study responded to exenatide. Some people did and some people didn't. So there's a lot of research going on as to why some people were responding to exenatide and others didn't. It suggests that there are different types of Parkinson's. And if you go to any support group, you look around the room, you will see that there are some people that are tremor dominant versus people that are more, have got more rigidity issues. So there appears to be different types of Parkinson's. We're not dealing with a singular condition, we might be dealing with some sort of syndrome. But my second answer to this question is that I don't expect any of these treatments to work. Now, I have to explain myself before you turn off your computer. We shouldn't go into any of these clinical trials with any expectations. These clinical trials are an unbiased evaluation of a drug or a treatment. And if we go in with any sort of expectation, then we are biased. So we must control our expectations at the start and even at the end of these clinical trials, and give all of these therapies the best possible evaluation that we can. So I have no expectations. I have high hopes, but I don't have any expectations. So my advice to everybody who's watching this is to keep calm and to lower your expectations. The lower your expectations, the happier you are in life. I'm a cup is completely empty kind of guy when it comes to expectations. The sun came up today and it was fantastic. But this is where individuals like Martin Taylor come into the picture. And he's very much of the, we must be very careful with regards to expectations mindset. And he likes to focus on positive realism. Martin was diagnosed with Parkinson's at age 32. And he has taken a very keen interest in the research that's going on for Parkinson's. He's set up the Facebook Parkinson's Research Interest Group. And he is encouraging a philosophy of positive realism. So, which I am completely beholden as well. Are there reasons for optimism? Yes, there certainly are reasons for optimism. If only if you look at this graph. In the 200 years that have passed since James Parkinson first described Parkinson's, there has been almost 140,000 research reports involving this condition. But it's only within the last 20 years that we've seen the bulk of those. 80% of them have been published in the last 20 years. And there's so much research going on at the moment that there are certainly reasons for optimism. And the research is going in so many different directions as well. Instead of just simply being an L-DOPA, a different type of L-DOPA that's being tested, we're seeing exenatide, we're seeing anti-inflammatory approaches, we're seeing stem cells, we're seeing so many different approaches being applied. There is certainly reason for optimism. How can you get involved? There are multiple ways you can get involved with the research. Even if you don't understand anything I've said today, you can certainly be involved with regards to the communication of not necessarily the research, but the experience of having Parkinson's. So I encourage people watching this to get involved with the research. It's a very exciting time for Parkinson's research. This leads me to one of my favorite individuals in this world. I've never actually met Mike, but he's a hero of mine. Mike Lloyd, he's a fellow Kiwi, and Mike was diagnosed with Parkinson's 9 years ago. And last year, he actually ran his 10th New York Marathon, which is quite remarkable for someone who's 9 years into a diagnosis with Parkinson's. What's even more remarkable about Mike is that Mike is blind. And for Mike, it's not about what you can't do, it's only about what you can do. So it's a celebration of what he can do as opposed to what he can't do. And people ask him, Mike, what are you going to do when your Parkinson's is to the point where you can't run anymore? And he says, well, I'll find something else to do. So it's not about what you can't do, it's only about what you can do. And with that, folks, I will say thank you very much, and I'll be happy to take any questions.
That was a wonderful talk. I was quite interested when you sent me the slides originally in the title of the talk, and it was good that you explained it so well. I thought also that there was a tremendous amount of hope within the presentation. I know that drug trials are very complicated, but it was good to see that there are so many different avenues being explored at the same time.
And only a small fraction of the avenues being explored at the moment was discussed there. There's things like LRRK2 inhibition, or there's just so many different avenues being explored. It's bewildering as a researcher, but it's certainly reason for optimism.
Certainly, yeah. Do we have questions for Simon and Liz?
I do. I have a few in the chat box. One from Helen. How on earth did scientists find out that a reptile's venom has molecules in it that could treat people? And is any research being done to find other useful molecules elsewhere in the animal kingdom?
Yeah, it's a really fascinating story. The gentleman, I forget his name now, who he won there's a prize called the Goose Award in science, and the Golden Goose Award, and he actually won it. I don't know if Heather or David can remember the discoverer of xenotide, but researchers are looking under every stone. There's lots of fascinating stories with regards to where a lot of our medication came from. One comes from soil samples collected on Easter Island, for example. they divided Easter Island up into different quarters, different small segments, and they dug in the dirt and they grew bacteria, and there was about 40 different samples collected, and there was only one of these soil samples that actually gave a result, but the result has led to a drug that's now widely used for suppressing the immune system. So there are researchers all over the world digging under all sorts of rocks and in the deepest parts of the ocean and finding all sorts of fascinating bits of biology, and then seeking to apply them to medical uses. Sorry, what's that question?
It was Dr. John Enns who discovered it.
Yeah. Yes.
No, I think that covered it. She asked, how someone discovered that the reptile venom was useful, and what else is being done to look at animal kingdom potential solutions. A question about. Oh, sorry. Go ahead.
I was just going to say, researchers are looking everywhere.
Thank you. Another question about exenatide. If this drug comes to clinic for people with Parkinson's, is it something that would be prescribed only for people at the time of diagnosis to slow progression, or could it be for anyone at any stage of Parkinson's?
It's probably too early to say at this point. What we know is that a subset of people are responding to it. We don't know if it is a disease-modifying effect. This also needs to be better explored in this phase 3 trial. It's still too early days to sort of give any indication.
Thanks, Simon. And G-case linked to Parkinson's, what sort of symptoms does it cause that this drug would reduce or improve?
So G-case is produced by a segment of our DNA, a region of our DNA, a gene in our DNA called GBA1, and mutations in this gene increase people's risk of developing Parkinson's. And these folks who've got GBA-associated Parkinson's typically have a faster progression in their symptoms of Parkinson's, an earlier onset or earlier diagnosis, date of diagnosis. And with regards to the approaches that are being used to target G-case, we don't know yet if simply boosting G-case levels or replacing G-case will slow down or turn around Parkinson's. We're not sure yet. This is again something that's still being investigated. But ideally, what's really interesting in the situation is that people without GBA-associated Parkinson's, so without a genetic variation in their GBA gene, these individuals, there's quite a lot of people with idiopathic Parkinson's who also have very low levels of G-case. And so it might not simply be that the subpopulation of Parkinson's with this particular genetic form will benefit from drugs like ambroxol. It could be that people without a genetic risk factor for GBA might also be benefiting if it is found that raising G-case levels and G-case activity have a beneficial effect on Parkinson's symptoms in general, or progression in general.
Thank you. I think that's all the questions from the chat box, aside from how can you pack so much in so clearly to a very complicated talk. So perhaps next time we could have some public speaking coaching.
I'm just flattered that people are still here and not haven't wandered off. I thought I was brushing a bit, trying to stay to time, but thank you.
That's all the questions from the chat box, John, that are specifically for Simon. So we could, I'll hand back over to you. I do have one more question for the panel Q&A segment.
Okay. Thank you very much indeed, Simon. That was a wonderful talk, and thank you very much for answering all our questions. I'm now going to hopefully share my screen. I seem to have a problem with this each time. So now it's time to invite our panel of experts to answer your questions. So do we have questions, Liz, for the whole panel?
Yes, I have one question from the chat box from another John Telford. He asks, a major reason for a drug repurposing approach is that because the drug is already approved for another condition, it would not have to go through some of the lengthy processes to be approved for use in Parkinson's if it's effective. But if a drug is chemically altered, for example, to remove some of the adverse side effects, surely it has to be regarded as a new drug and would therefore lose that advantage? Question mark.
Maybe I can kick off with that one, Liz, as it's something I specifically referred to in the talk that we had done as part of one of the studies. And essentially the answer is yes. If the drug is chemically modified, then even if it is based upon a drug which has gone through the process and is licensed, if it's chemically modified and therefore a new chemical entity, then it does lose those advantages of speed, and it still has to go through the safety and toxicology testing.
Thanks, Heather. Any, Simon or David, anything to add?
This is just one other thing on repurposing, because the thing is that often these drugs are off patent and they're produced by companies which manufacture what's called generic drugs. So even if these drugs are positive in trials, there's a big challenge in actually getting them to patients. So what the NHS have done is actually develop a new scheme that they've been talking to us about, is because say for ondansetron, which is in one of the trials funded currently by Parkinson's UK, there are 8 manufacturers of ondansetron. So who actually then takes it to the regulators? So the NHS are saying that they've got this new scheme that they're happy to work with people who've got a repurposing candidate, and they will actually pay for the regulatory approval of this drug, and then they'll share the regulatory license across all the current manufacturers in the UK. So there is now a pathway. So if we do identify a repurposing candidate, there is a pathway to get these drugs into the NHS. So that's worth just another point making as well.
That's really encouraging news, David.
I have another question, John, from Stephen. For the panel, regarding the trajectory, the period between initial signs and referral for Parkinson's is very varied depending on your GP's awareness. Is there any operational research looking at how this might be improved? So I'm assuming this is primarily a question around better and earlier diagnosis in Parkinson's.
So I could probably kick off on that. So there is a study ongoing at the moment, which is called Predict PD. And this is all about trying to actually develop an algorithm so that we can better spot people who are actually developing Parkinson's. So this is still ongoing. They're looking to recruit 10,000 healthy individuals who are between the ages of 60 and 80. And so they're looking for the very early signs of Parkinson's before you actually go and see a GP, say about a frozen shoulder or a slowness of movement, to try and identify ways in which we can detect it early. So they'll send out smell test kits, They're going to start doing genetic testing. You can do tap tests on keyboards, and eventually we're hoping that you'll be able to generate an algorithm so that, you know, when you've gone to your GP with a certain number of symptoms, you know, a red light will flash up on their screen, because they're constantly on the screen at the moment rather than actually looking at you as somebody, you know, in their clinic. It'll flash up from saying, you know, have you considered this person might have Parkinson's? So hopefully one day we'll actually be able to, you know, get some tools that will actually help us diagnose people with Parkinson's a lot earlier and a lot more accurately as well.
I think that will be very important, because I think there's evidence that the sooner you begin your treatment, the better your chances of retaining your movement and not having so many symptoms over time.
I have another question here, John, for the panel from Joe. Why are pharmaceutical companies investing less in Parkinson's?
I can probably have a shot at this one. Tough question, but typically pharmaceutical companies are not the ones to watch for with regards to the development of new drugs. It's more the biotech companies that come through. The university researcher will discover something in the lab. They'll start up a biotech company. The biotech company eventually gets grabbed by a large pharmaceutical company once the biotech company has demonstrated in phase 1 and phase 2 that the drug is safe and doing something. And you're seeing this now with a number of different companies. For example, there's a company called Inflazome, which is an Irish company that's just been bought by Pfizer. They've got an anti-inflammatory approach for Parkinson's. Excuse me, Roche, not Pfizer, Roche. It's been purchased by Roche. And so this is where Roche is saying that we think this is very interesting for Parkinson's and we will take it forward. And then you've got other companies, for example, Denali. They've just signed a large collaboration with the pharmaceutical company Biogen. There's a bit of a misnomer that's with regards to LRRK2 inhibitors that Denali has been developing for Parkinson's. There's a bit of a misnomer that the pharmaceutical companies have run away from Parkinson's. It's not necessarily true. Pfizer stepped back and they made a bit of a PR announcement about stepping away from neurodegeneration. But at the same time, they set up a large fund for research, for funding research in neurodegeneration and Parkinson's. And they also spun off a small company to look after a lot of the assets that they've been working on. So it's not necessarily true that pharmaceutical companies are scared of neurodegeneration or Parkinson's, or that they're avoiding it and they're not spending money. They are spending. They can see that there's enormous, this is going to be an enormous societal problem going forward as our population, the Western populations, age. But they're being very, very cautious about how they approach. They're placing very careful bets, and they're waiting to see. They're waiting to see the data. So they're letting biotech companies develop the new treatments, and then they're grabbing them at the later stages of development.
And Simon, I'll just quickly comment as well on that. So another way in which the big pharmaceutical companies are working is, rather than having their own in-house research labs working on Parkinson's, let's say, they're actually creating quite long-term research collaboration agreements with academic labs that specialize in Parkinson's research. And so that's one way that they're continuing to work in the area without having the actual physical facilities and research themselves. And then obviously they can fast track or let go various targets that are investigated through those academic industry collaborations. And Sheffield has a lot of those. We have a lot of those at SITraN, and a lot of other UK universities and actually international universities have those sometimes quite substantial and long running industry partnerships and collaborations.
Thank you. I have a question here from Helen. Do we have any updates on the lady who can smell Parkinson's? I believe she's referring to Joy Milne, the nurse in Scotland. And a follow-up study, I believe, was being carried out by, was it Perdita Barron? Helen also says, I heard that dogs can potentially smell coronavirus. Is this a potential avenue for Parkinson's diagnosis?
Yeah, perhaps I can. So it started off from a bit of research that was funded by Parkinson's UK. So then it went more on a laboratory-based situation. So they actually took swabs off the skin. And so then they found 3 components in sort of the sweat from the skin that was actually indicative. So supporting the reasons why she could actually smell people who have Parkinson's. So this is actually being taken forward as a diagnostic test. So now they're trying to look to see whether it's specific to Parkinson's or whether it's common to Parkinson's-like disorders like MSA and PSP. But also they're going to try and look in other neurodegenerative disorders like Alzheimer's to see whether we can actually roll that out as a diagnostic test. So work is carrying on that.
Thank you, David. And another question about sort of early diagnosis from Alan. Alan says he briefly met Simon at a clinic open day at Cambridge some years back. And at that time, Simon was working on using people's breath as a means of detecting Parkinson's, perhaps linked to an Israeli company. And did anything come from that research?
Yeah, so the manuscript for the study is finished now, and the manuscript for that is in its final form, being written up and about to be submitted for publication. And we'll be drafting an information sheet for the participants as well. What we are seeing is that we can certainly differentiate between people with Parkinson's and without Parkinson's, and we can also see differences in the levels of progression. This was a study that was funded by Parkinson's UK and BIREX, which is the British Council and Israeli collaboration, and some Israeli engineers at Technion have developed a method of examining the molecules in breath samples, and they're applying it to all sorts of different medical conditions. And primarily, initially it was lung conditions and gut conditions, and then they sort of started to spread their interest more broadly, and Parkinson's popped up. And Professor Roger Barker in Cambridge, where I was working at the time, initiated collaboration with them. And as I say, the publication, the manuscript of the results is in the works at the moment.
Thank you. That's it from the chat box. John, if you have any questions.
I've got 1 or 2. How does aerobic exercise benefit people with Parkinson's exactly? What is the scientific basis? Martin asked this question, and he asked it particularly because after a hard spinning session, my tremor is more pronounced for an hour or 2, but hopefully progression is reduced.
Yes, perhaps I can start off with that one. So there's been a number of studies. There's been quite a lot of animal research. So there's, as we talked about before, there are some genetic forms of Parkinson's. And so one of the genetic animal models, if you induce that in mice, and mice normally like to run in their wheels, in their cages. So if you let mice run in their wheels, and then in another population of mice, you prevent them from running and they're quite sedentary, the actual spread of the bad proteins that cause Parkinson's in these animals is actually greater in the animals that don't exercise. So also there's been some brain imaging studies in humans where they've actually had groups of people who exercise, and then they put them in a brain scanner. You can actually see more dopamine receptors in the brain, so you'd be more responsive to what dopamine is being released from your nerve endings, but also from your drugs as well. So exercise is also important in a process called glymphatics. So this is a new area of research. And so Parkinson's UK have done a joint fellowship with the Alzheimer's Society on this. So this explains about when you sleep. This is why sleep's important, because it's the time when you drain the bad proteins out of your brain. But they've also shown that exercise increases this glymphatic process. So there's lots of evidence which is gathering now exactly what exercise stimulates causes effects we don't know. So there's lots of things floating around in your bloodstream after exercise. You also get the euphoric effect because it releases endorphins into your bloodstream. But exactly what exercise does on a chemical basis in your blood to trigger something in your brain to slow Parkinson's, we're not anywhere close to that. But because then you could actually think about, well, if we can identify what component, what is exercise doing to trigger these effects in your brain, you know, perhaps we could actually develop that as an alternative therapy.
Thank you, David. Simon and Heather, do you have any other comments to add to that? Okay. Well, I've personally benefited from exercise. I've been assigned to the Parkinson's Warrior exercise program not long after I was diagnosed, and I certainly found that tremendously helpful. And so I would encourage people to take as much exercise as they feel able to do who have Parkinson's, because I think it's generally recognised that exercise is beneficial to everyone in society, whether or not we have a condition or are healthy ourselves. I've got a general question. How has the COVID-19 pandemic affected research in the UK globally, or has it presented any opportunities for researchers?
Heather, do you want to speak from a personal perspective as a researcher?
So it's obviously presented a lot of challenges in terms of continuing research. In March, when the national lockdown in the UK took place, and also then in other months around the world when various national lockdowns were happening, then pretty much all laboratory, certainly academic laboratory facilities, were closed to all but absolutely essential work. And so that may have meant keeping the animal facilities, for example, running if any of the studies were involving animals, but even cells from patients or people with Parkinson's that we had been working on in the laboratory for the previous 6 months, going in every day to feed them, they had to be thrown away. So from that point of view, the lockdown had quite a large effect because then subsequently the laboratories were shut and people were working at home. And obviously if it's laboratory work you're doing, then you can't do that in your kitchen. However, what it did mean was that people had more time at that point for complex data analysis, for refining data analysis to speed up that process, for catching up on literature, which is often difficult to do when there's conflicting demands in the laboratory. And I know a lot of people have made use of that time away from the lab very effectively to enable their studies to actually run quicker when they can get back into the laboratory. How it's running now is that most laboratories are open, all but at a more limited capacity, but work has been restarted for the last few months and is now continuing and picking up pace. I would just add one final thing in that a large area of research which has kind of come up from COVID is the effect of COVID on developing Parkinson's, and that's actually led to some very interesting and novel collaborations both nationally and internationally into that particular area of research. So I think there's the obvious cons, if you like, of COVID, but there are actually also some positives that you can take from it.
So there's another point to this, is that, as you probably know, that during the COVID pandemic, you probably heard drugs being, going through into clinical trials within a matter of days between the applications going in. They were rapidly approved for going ahead in clinical trials. So what we've been saying to the government is to learn from the experience of this accelerated transition from the bench to the bedside, and actually start applying it for general applications, because at the moment it's a very long-winded process to get your application approved to get a drug into patients in a clinical trial. So we're saying to the government, take notice from the COVID experience and try and accelerate the process up, because at the moment it can take you, probably 6 months to get an approval for a clinical trial. So why can't we get it approved in a bit shorter time? Not quite as quick as a COVID drug, but we would like it perhaps done in a couple of months rather than 6 months. Oh, and just one other point on COVID. We're just about to announce that we're doing a whole series of research. So this is headed up by Camille Carroll. So this funding will actually allow 20 NHS trusts, and hopefully we'll get more to join, to actually do some in-depth analysis about whether people affected by Parkinson's are actually indeed at more risk of developing COVID, or is it simply your increased risk is down to having more of an advanced age, or whether you've got underlying diabetes or another condition. And also the research will cover whether, if you're admitted to hospital, whether your journey is any different to anybody else who's admitted to hospital with COVID complications. So then that will actually give us some hard sort of evidence, to tell people about whether they are actually at increased risk or not. So that work, hopefully we'll try and get that done as quickly as possible and released to everybody, and do some publications on that as well.
Thanks, David. There's a related question in the chat box from Robert. He asks, is anyone collecting data from people with COVID who have had smell and taste problems? He comments that people who had Spanish flu were traced and went on to develop Parkinson's much later in life. Do we know much more about that at this stage? It's a tough one.
Yes, I was going to say.
I did share a link to our recent research blog post on the subject in the chat box too.
Yeah, so there is, because the Spanish flu, it killed 52 million people worldwide, and as already mentioned, a number of people who survived the flu actually went on to develop a form of Parkinson's later on. So there is a big concern at the moment whether having COVID actually renders you more susceptible to developing neurodegenerative conditions later on in life. And there is quite a lot of evidence to support that you are seeing some changes in the brain, as you say, you've already got loss of smell and sense of taste as well, but whether that actually translates to increased risk. So there is some research being done. I think there's a move to try and get that done on a national level across the UK, so to try and monitor people long term.
Thank you very much, David. Do we have any more questions, Liz, or have we covered everything?
I think there's one final question in the box, and this is probably the last one we have time for. Actually, I think we're running a little bit over. But the question is, Helen is very interested in the links between Parkinson's and dementia. Are there any studies or research being done specifically in this issue that people should read more about?
Okay.
Have a shot of this. Oh, sorry, David, go ahead.
No, you go ahead, Simon.
One of the studies is focused on Parkinson's dementia cohort. So these are individuals with Parkinson's who are having cognitive impairments. And there are other clinical trials also. The Ambroxol study that's being conducted in Canada is also focused on individuals with Parkinson's and Parkinson's dementia. So there are clinical trials ongoing for a lot of the drugs that have been discussed today, focus on these individuals.
Yeah, just one thing to add there, that if you look at the brains coming into the Parkinson's UK Brain Bank, there are, well, it's quite rare to find a Parkinson's case that doesn't have some Alzheimer's type pathology in it. But that ranges all the way up to where they get severe Alzheimer's pathology in them. And so these tend to be the ones who develop cognitive decline. So certainly there is a crossover between Alzheimer's and Parkinson's, and certainly there's a lot of interest about whether drugs, developed to treat Alzheimer's would actually have a benefit for actually slowing cognitive problems in Parkinson's. In fact, one of the trials that I mentioned, PD Mind, which is an IMI program one, that is actually looking at a drug which was originally developed in the Alzheimer's program to see whether it would slow down cognitive problems in Parkinson's.
Thank you, David. Well, I think I'm going to bring the question and answer panel session to an end now, if that's okay with everybody. May I thank our distinguished speakers, David Dexter, Heather Mortiboys and Simon Stott, for their excellent presentations and for being with us all morning and answering so many questions, some of which were, I think, pretty testing. Thank you very much. I've got a few acknowledgements I would like to make, if that's okay. The event continues to be subsidised by local branches and Parkinson's UK. The Parkinson's UK East Midlands Research Interest Group is very grateful for funding for this forum from Leicester and Nottingham branches of Parkinson's UK, as well as money given in memoriam from some special people from the Derby branch who had an interest in research. I'd like to try and share my screen with you just for a moment. So I'd like to thank all the Pembreg team for their hard work in organising this event, especially Liz Nash for her technical support and for handling the questions so adeptly today. I'd also like to thank Steve Johns and Ian Bilcliffe, Chair of Nottingham and Derby branches respectively, for their involvement and input and their support. I'd like to thank our three distinguished speakers for their presentations and answering so many of your questions. And I'd also like to thank you all for your questions and for registering for the event and for helping make it such a success by your interest in research. It was particularly good to have so many of your questions, so we felt you've been involved all the way through. I'd like to ask you to complete an evaluation which will be sent out this afternoon to all of you. This will help us plan and improve and learn from today's event. I'd now like to ask everyone to show your appreciation for our speakers in the usual way with a round of muted applause. We've certainly had some very positive messages come through the chat box, and I know that people have really enjoyed the session. So my thanks to everybody involved. Thank you very much indeed. I now call the meeting closed. Thank you and goodbye.
Thanks, John.